So we will now continue with our session, and the first speaker here in person will be Anna Maria FARSAKOGLOU from University of the Aristotle University of Thessaloniki, and the floor is yours.
Okay, hello everyone, and I'm really glad that I'm here among you and that I have this opportunity to present you some first insights of the genetic diversity and differentiation of 12 major European tree species. So to begin with, the major European conservation threats are the human activities and habitat loss, the land use change, and of course climate change, or as the topic of this conference says, a changing world. So how we're going to fight against the climate change? Well, what a 16-year-old girl does is to do school strikes for climate change every Friday. But we, the scientists, we do have a different approach. So what we do is we go out on the field, then we do some lab work, and then we do some endless scripting in order to analyze our data. So in GENTREE, we have 12 species in total. So we have 7 species and 5 species that we have used 2 different methodologies to analyze them. And this is because when the project was designed, we had more genetic information on the 7 species and less on the 5 species on the left. So we used the exome capture sequencing for the 7 species and the single primer enrichment technology for the 5 species. So the project design was, for the 7 species, we had around 24 populations in pairs per site for each species, and around 10 populations per species and per site. So for the SPET technology, I will tell you in brief what each technology does. So some probes were designed, and we target some SNPs that are known for the high levels of polymorphism. So we targeted some SNPs, and close to them we also managed to get some novel SNPs, around 2 or 3 per region. And also for the 7 species and the exome capture sequencing, we followed a different approach. So it's an alternative to the whole genome sequencing. And we have managed to have the exome and approximately 1 to 2% of the whole genome. So for those 2 methodologies, the SPET methodology targets specific SNPs in advance. And we also detect some non-target or novel SNPs. And for the exome capture sequencing, we captured orthodox genes, and all the SNPs are based on de novo assembly. Then, to construct our data sets, we did some, I would say, intense filtering. So for the moment, we have used only the targeted SNPs for the SPET data. We removed triallelic SNPs and complex variants. We have moved also SNPs that have more than 50% of missing data. And we have chosen 1 SNP per condict based on the SNPs that have the lowest percentage of missing data. And we did that 1 SNP per condict selection because we wanted to reduce the bias due to linkage disequilibrium. And a similar approach for the exome capture sequencing, we excluded newly generated invariant sites and also SNPs that have more than 50% of missing data. And here we also select 1 population per pair because we want it to be uniform and consistent among the 2 methodologies. And also for the same reason, to reduce the bias due to linkage disequilibrium, we selected 1 SNP per condict again. So here we have some numbers of the SNPs of all the species. So for the exome capture sequencing, we have started from around 100,000 SNPs, and we have ended up with 10,000 SNPs. And for the SPET data, we have started from 10,000 SNPs, and we ended up with around 1,000 SNPs. And then it was time for our SNP analysis. So we have managed to construct bioinformatics pipeline, which I will be happy to explain you later at the coffee break, because it's a poster. If you haven't seen it, I will be glad to explain you more about that. And now I'm going to give you some extremely preliminary results. I'm going to use some examples from some of the species, of the 12 species, and I'm going to talk about the expected heterozygosity and the FSTI, which is, in our case, it's not the normal FST, but is when we compare the genetic differentiation of one population compared to all the other populations. So here we have the Aleppo-Pine data, the estimates. So we have the expected heterozygosity, and we can see that we have higher levels of heterozygosity on the eastern part and higher levels of genetic differentiation on the western part of the distribution. For Taxus baccata, we have used the expected heterozygosity as a deviation from the mean, because the results are not, they don't have a pattern across Europe really. So we have high heterozygosity levels from Greece, Central Europe, and Norway, and higher genetic differentiation on the western part. And from the exome capture sequencing data, we have higher heterozygosity levels in the north and higher genetic differentiation in the south. And for Fago sylvatica, we can see a decline of the heterozygosity starting from higher heterozygosity in the southeastern part of Europe, and it declines up to the northwestern part. So, for the two methods, of course, we have higher heterozygosity levels in the SPET data, because the SNP selection was done due to the high polymorphism that we detect on those targeted SNPs. And the exome capture sequencing has lower heterozygosity levels, as the genome areas that were chosen, the SNPs were chosen in random. So, what we can get from those data, actually, in the conservation sense? We use two different genomic techniques and target regions of interest, and to elucidate conservation needs and conservation threats. And thus, we can presumably characterize some new gene conservation units and also some important European gene pools. And also, we can detect some vulnerable populations that are in need of conservation. Also, we can identify SNPs that are significantly associated with the environment and some prospects for local adaptation studies contributing to conservation. The next step is to analyze adaptive versus neutral diversity, and to promote the development of useful tools and strategies for conservation and management. And I also want to say that it's very important that we had all those kind of approaches in those data, because we had, for the 12 species, the same sampling design, the same methodologies for analysis, and so we have like sort of a uniformity and consistency across all the species. So how the conservation can be implemented in forest management? We can select populations for conservation. And then, as conservation is part of the evolutionary management, we can ensure the adaptation of forests and forestry into new conditions. And with that, I would like to thank a lot for this opportunity that I have, and to thank my supervisor, Phil Ravanopoulos, And also I would like to thank Bruno and Ivan, Scotty, that they helped me throughout all these months, and the whole consortium that I think we do a really, really good work, and we have passed a lot through all these years. And you for being here today after the lunch, which is a hard time, I know. Thank you very much.
Hi, Pascal Milisi, Uppsala University. You said that you picked just one SNP per conjugate to avoid linkage disequilibrium, right? For the de novo assembly, do you have an idea about the distribution of the size of the conjugates?
Well, I know that the exome capture sequencing has like big lengths. Probably you know better than me. But the SPET data were really close to each other. So we were thinking that they're linked to each other.
So the size of the conjugates for each species varied a lot within a species, but it varied also a lot between species. The fact to take a SNP on two different conjugates does not ensure you are told that there is no linkage disequilibrium between them, because sometimes it is considering as being two different conjugates just because a small part in between the different targets was not grasped in the experiment.
Yeah, that's true, but we wanted to reduce it as much as possible. I mean, to reduce the bias.
In that case, you can go for statistical linkage disequilibrium analysis to remove the SNPs that are into high linkage disequilibrium.
Yeah, this is like a preliminary analysis that we have done, so we will reconsider everything and
yeah, sure, but
we'll do more.
We can enjoy that time to discuss this also, so that it can be faster later on, right?
Sure.
Thank you.
Yeah, thank you.
I would like to follow up on this last point that you made, how this can be then used actually in conservation practice. So do you see a large mismatch between what is currently the conservation practice and what you would suggest based on your data? I mean, is there a change needed, or is it just filling information into the awareness of the people who are responsible?
Well, I think it's too early to make any conclusion out of this data, but I think they are very promising.